for the final.
Well, thank you very much, Art. That was very nice. It's been really a wonderful treat for me to be able to come to this meeting. It's been so interesting and, for the most part, so entertaining that it's something that I would normally never have heard about. I work, as you've heard, on a subject really entirely different from all the other speakers and from most of the people in the audience, I think. So we, as Art said, are interested in peptide chemistry and protein chemistry and biochemistry. And that's what I want to talk about today. I started, I guess, about in the mid-1950s with a peptide growth factor that we had isolated, and then it came time to synthesize it. So that was my first experience with trying to make a peptide, and after many months I finally made this little pentapeptide. So the existing methods worked, but they were very time-consuming and labor-consuming, and it seemed to me that if we were going to make larger numbers of peptides, and particularly longer, more complicated peptides, we needed some new methodology. And I kind of sat on that idea for some time, and then one day it occurred to me how we ought to be able to do that. So that's what I want to try to describe today, and then some applications from our lab and from other labs. So if we can have the first slide. See if I can get everything together. Okay, this pentapeptide was made by classic methods that were developed originally by Emil Fischer in 1900 and were used by everyone up until this time. When I'm sideways, can you hear me? I have to talk right into it. Okay.
Somebody would turn that podium about 90 degrees, it would work.
That would help, yeah. Anyway, here is an outline of the approach that people take. It's called a fragment or a segment approach. If you want to make a peptide, then you start with the individual amino acids, and for example, put 2 together to make a dipeptide, put 2 more together to make another dipeptide. Can we move that around? Oh, that's better.
Yeah, somebody had a podium.
Okay, so now you have your two units here, and then you join them and make a tetrapeptide, and the same thing over here, some combination, and finally put these 2 together to form the full compound that you're trying to make. Now what you had to do, you carry these reactions out in solution. All the reagents are soluble and the reactions occur in solution. You have to, after each step, for example, you make this dipeptide, you have to isolate it and analyze it, characterize it, purify it, and then go ahead with the next step. Well, that's okay for a few steps, but if you want to make something large, then it would just take you forever. So that's what I was trying to do, is find a better way to do that. The next slide is an outline of the idea. Oh, I have this here, sorry. This is an outline of the method that I had in mind. It only took a few minutes to think of this, but it took me 3 years before I could make this idea work. So it took a long time to develop it. So if I could go through it briefly, we start with a solid particle, some resin, and put a functional group onto it, so it's reactive. Then take our first amino acid and attach it, and this is a covalent stable bond here. Then you remove a temporary protecting group, and you can add the next monomer unit, another amino acid, to make a dipeptide. And so on. You keep adding the units until you assemble the peptide chain that you're interested in. Then finally, the stable bond here, which is usually an ester, can be cleaved under more rigorous conditions. That liberates your product into solution, and then you purify it and analyze it. So for all of the intermediate steps, the purification was simply washing to remove the excess reagents and the soluble byproducts. See, the resin is selected because it's completely insoluble. It's solvated, but it's insoluble. So the peptide chain that's attached to it is also insoluble. Therefore, you can filter and wash without losing any of your material. We had to select each of these variables, that is the resin, the functional group, and the coupling reagents, and so on, had to be examined individually and try to find a group of reactions that would go together and allow us to carry out this scheme. The next slide shows the resin support that I forget that I'm running this. What we finally settled on after examining a number of solid supports was a polystyrene up here, which is cross-linked lightly with divinyl benzene. So these linear chains then become insoluble because of the cross-linking. And they were in the form of small spheres shown below, which we originally obtained from Dow Chemical Company. And they're something like 50 microns in diameter. The next slide shows I can't seem to learn that. Try that again.
Are you going back?
Oh, no, I skipped one though. Huh. I don't know. There's one missing, but I don't have to have that, I suppose. It's a scanning electron micrograph of the beads, and just shows them to be nice little spheres. See where we are now. Well, okay. So now we have the resin support that would work. Now we had to use it and work out the chemistry. Well, a lot of different chemistry will work, but it took a long time to get the first one that would work, and that's what this shows. So we just briefly, you take the resin here, which has aromatic rings in it because of the polystyrene, and you take a chloromethyl methyl ether and a zinc chloride catalyst, and you can make a chloromethyl derivative. That's like a benzyl chloride, so it's fairly reactive. Then you can take the first amino acid protected on nitrogen and a cesium salt, and that will form then an ester, a benzyl ester. Now this Bach, which is a tertiary butyl oxycarbonyl derivative, can be removed fairly easily with HCl or with trifluoroacetic acid. That gives you the protonated amine. Then you wash with triethylamine and you get the free NH2. Now it will act as a nucleophile for the next reaction, which will be with another amino acid in which the carboxyl group is activated. And that also can be done in many different ways. We often use dicyclohexylcarbodiamide. So that activates a carboxyl, so you have a leaving group, and then the amine comes in and attacks that. That forms the amide bond, or the peptide bond. Now you simply go back and repeat the deprotection, the neutralization, and the coupling to add the third amino acid, and so on. Finally, though, with a very strong acid, hydrogen fluoride, anhydrous, this benzyl ester can be cleaved, and the Bach group, and that gives you then the free peptide, which has to be purified and characterized. So this is a general scheme, and lots of variations of it have been worked out, some in our lab and some in many other labs around the world. This is the difference of opinion about which ones are best. It depends on who developed it, which one he thinks is better than somebody else's. But I think that's enough of the detailed chemistry. This all took a long time, and then we spent another 10 years with looking at each of these variables in more detail. The reason this thing works, as I mentioned before, is the property of the resin itself. It's not just a solid hard lump, but it's readily solvated. Well, I seem to be unable to do this. Can I just ask for the next slide then? I don't know what's wrong. Okay, so this is a sketch that I made just to try to illustrate this bead. So if you take a bead, little one, and expand it 500,000 times, then you get something that would look like this, a great huge thing. And inside then are the polystyrene chains, and on the chains then we're going to add the first amino acid and then grow the peptide there. Now it's not just one chain of peptide, but millions of chains are going to be on this one bead. But the point is the polystyrene solvates in organic solvent where you do the reactions, and it will swell. We have examples where it swells about 28 times its volume, so it gets much bigger. And on this scale that I have here, an amino acid would just be a pinpoint, and even a protein, small protein crystal, would only be so big. So there's room inside to accommodate a large molecule and to synthesize it and then get it out. And that, of course, is critical. There are other polymers like polyacrylamide, polyethylene glycol, and so on that have also been developed that will accomplish similar things. I'll try this one more time and then give up. Oh, okay. The peptide chains are uniformly distributed throughout one of these beads, not on the surface, but throughout the interior matrix of the polymer. And this is an autoradiogram where we made a peptide that was labeled with tritium. Then you embed the bead and slice it on a microtome, so you get a thin slice, and then put it on film, and everywhere the peptide was, you have a tritium, and everywhere the tritium is, you get a black spot. And the conclusion is that we have a relatively uniform distribution of these chains. And because of that, then there is no special crowding effect, and you can make a large molecule. The next slide. In order to I'm afraid the problem is some of these are very old slides now, and they're thicker than the usual ones that we have nowadays. So I do have some projector problems with them. I was just going to try to show a picture of a little reaction vessel that we use. Well, okay, it would be the previous slide, but anyway, the reaction, the chemical reaction, occurs in a little glass reaction vessel like this, which is on with a motor behind it. It's a shaking motor, so you mix the resin just by this rocking action, and that then allows the chemical reaction to occur. I do have to back up somehow here. Can you go back one? Well, okay. All right. Anyway, what I was going to tell you was that we had a little manual apparatus where we could carry out the reaction and decided to make a peptide hormone, which is something with some biological activity. So we selected bradykinin, which is a nonapeptide, and made it, and it worked out very well. The time scale was much shorter than the usual standard synthesis, and it had exactly the right biological activity in contracting smooth muscle and so on. So we made that, and it was at that point that I finally decided that our new method probably will work, and was anxious then to go ahead and try to develop it. If you want to make a large number of analogs, derivatives of such a hormone, you're going to want to find some way to make many more peptides than what I'm describing, which is making them one at a time. And later on I was going to mention new techniques where you can make something like 1 million peptides all at once and assay them and identify the one that has the best activity. So I'll mention that again in a minute. So having made bradykinin, my colleague John Stewart had been working with bradykinin, and he had made 3 analogs in a 1-year period, and he immediately went over to this method, and he made about 50 the next year. So it certainly accelerated the whole process. So then we wanted to extend it to something bigger to really test the methodology, and decided on a real protein to see if we could do that. And in order to try to make the protein, we had to mechanize. So we took our little reaction vessel, which was operated manually, and built this little machine. So we had in it we had some a pump and valves, selector valves, and reagents, and more reagents that were up outside of here. And the whole thing was controlled by a stepping drum programmer. So it's an electromechanical device. Nowadays these are made commercially, and of course they all have a computer in them, but they do roughly the same thing as this did. And we had a set of timers which control the reactions going on over here. So this machine still works. We have one, and 2 or 3 other people around the world have one, but the commercial ones have taken over because they had real engineers making them. And so we picked ribonuclease, which is the next slide. Well, this is a disaster. None of those slides will go down, I guess. Okay, we can go here. This is just another kind of a synthesizer where instead of a batch process, it's a continuous flow. So you put the resin in a column, and then you pump reagents through here and carry out the reaction that way. So this is a flow system, whereas our other one was a batch system. And there are all kinds of other variations of this, where you put the resin on a belt, and you run the belt through solutions and so on. So with the mechanized system, then we could look at the protein. The next slide then will show what we picked. All right, this is ribonuclease. The sequence had been worked out by Moore and Stein, and the crystal structure by Richards, and this is a model. Well, what it shows is the position of every atom in the molecule, and it's 124 amino acids, so there are lots of atoms in a fixed position. And when you look at this, you wonder, well, how can I go in the lab and put these things together so that every atom is in the right place? And of course that's overwhelming. You don't know what to do. So we have to look at it in 2 dimensions, and the next slide shows that. well, maybe it won't show it. Anyway, you can draw a two-dimensional view because the units then, instead of atoms, will be amino acids. And you have 124 amino acids all lined up. And too bad. So this next picture would be the one of the structure that Moore and Stein had developed. Well, okay. Can we try one more time?
The slides are metal and they're just too wide to fit.
Yeah, well, I realize that, but I thought they'd probably fit. Okay, well, this is okay. We'll go to here. So in white, you can see the two-dimensional structure, and even that was an enormous jump for us from our little nonapeptide to go up to 124 amino acids. So we decided, well, we'll have a safety project. Bernd Guta came over from Germany as a postdoc, and I was a little afraid if he tried to make the whole molecule, it wouldn't work, and he'd be left with nothing. So we had a safety catch, and we decided to make only the C-terminal peptide, which you can't see is in red, and what we would do is inactivate this histidine with a carboxymethyl group. So the whole protein would be inactive. Then we made this peptide with just a histidine present. So this is only 14 amino acids. And the hope was that it would insert itself into the molecule non-covalently and regenerate activity. This had already been done by splitting at residue 20 up here enzymatically. Again, Richards had done that. So he could prepare this peptide and the rest of the protein, separate them, purify them, and they were both inactive. But when he mixed them together without reforming that bond, they generated full activity. So that's the reason we thought we might be able to do the same thing at the carboxyl end of the molecule. The trouble was we couldn't split this thing, so we tried this inactivation procedure, and that just didn't work. And we tried, could never do it. So we turned to the whole molecule again. The next slide shows that, I hope. Well, that's too bad. Anyhow, it shows our 124 residue protein with all the protecting groups that are present on it, and it was our target. And so Guta set about assembling that, and he eventually put it together in a relatively short time. And then he could purify it. And now let's try the next slide. That's better. And what this shows is simply the various steps over here on the left that were used in the purification. You had to assemble the chain. You had to cleave it from the resin support. Then we went through some chromatography and a tryptic digest and so on, and an ammonium sulfate precipitation. And this gives the yields. So you start with 100% of the first amino acid, and by doing all these different things, we ended up with about a 3% yield, which sounds low, but actually was not bad at all, and we were very happy to see that. The last column gives the specific activity of the molecule, and it finally ended up with 78% specific activity. So it was not 100% pure, obviously, but it was pretty high and beyond anything that people had done up to then. About 10 years later, Yojima in Japan made ribonuclease, and he had then affinity chromatography. So he also obtained about a 3% yield, but with the affinity chromatography, he was able to purify the thing and got it up to nearly 100% specific activity. This also, the molecule had the right substrate specificity, which was very important to us. And next slide will summarize that. Okay. Anyway, this, there it is. All right. This is done on a crude preparation, not on the final material. But what it shows is that RNA would serve as a substrate and DNA would not, which is what you expect. And that cyclic cytidine phosphate also was a substrate, whereas the guanine or adenine containing nucleotides were not substrates. So the synthetic material did do what it was supposed to do, and that was very satisfying. What you can say here is that we could finally, for the first time, start with individual amino acids, and you could assemble them in the lab to form a real protein that had real enzymatic activity. It also provided new evidence for the theory that Anfinsen had proposed, which said that the primary structure, primary sequence of the enzyme was responsible for the tertiary structure. So it automatically folded into the correct tertiary structure. His was based on inactivating the natural enzyme, and you had to always worry that it wasn't totally inactivated. This, of course, you could not have any memory of the correct structure because you never had it. So that it confirms his theory that the primary structure determined the tertiary structure. Well, now finally we went back. Let's try one more time. Okay, to the one where we had the C-terminal 14 residue peptide, but this time we removed 5 or 6 residues from the protein. Now, by doing that, this end of the molecule was much looser. It was not bound tightly to the rest of the molecule. Then the synthetic peptide would fit into position, and then you could regenerate the enzymatic activity of the protein. So finally, our little safety experiment finally worked. We could do one other thing along these lines. Can we try the next slide? See, this may be all of my thick ones, I think. So maybe we don't have to worry about that. This is just a different view of this experiment, where in red we have the C-terminal peptide, synthetic, but in addition we have in yellow the S-peptide, which is at the amino end, with this bond broken, and then in white is the rest of the protein. So you have 3 components. Each one contains one of the known essential amino acid residues, either a histidine at 119, or a histidine at 12, or a lysine at 41. So those were on 3 different molecules, and when you put those together without reforming covalent bond, this thing would refold and reactivate to give a high enzymatic activity. And that was something that was new at the time. It also gave us a chance synthetically to examine the various residues in the molecule. Theoretically, you could do that by the total synthesis, but that would have been very much more work. So by putting them into this smaller peptide at the amino end, we could then examine the role of valine and serine and aspartic in particular, and phenylalanine. And this aspartic acid, for example, you couldn't replace that with asparagine, with the amide, or with glutamic acid with one more methylene in it. So these structures really require an exact conformation to be active. With phenylalanine at 120, we were able to put in tyrosine and generate an enzyme that was more active than the parent one. Later on, discovered that giraffe ribonuclease, of all things, had a tyrosine and was more active than the bovine enzyme. So let's try the next slide now. Okay, so I wanted to go on now to more modern protein synthesis. A whole variety of proteins have now been synthesized. The HIV protease at 99 residues, for example, has been made by Kent and his group, and they were in fact able to crystallize it. So here you have a big synthetic molecule that you could get crystals out of, and that was an important step. And other people, maybe 20 other proteins are known which can be synthesized, and all by the stepwise method that I've been talking about, where you just add one amino acid at a time. Now what people would like to do is this so-called ligation method, which really reverts to that very first slide I showed, where you make segments and put them together instead of individual amino acids. And the other thing that they want to do is to use unprotected reagents, starting materials. There are lots of advantages, mainly for solubility reasons, for aggregation reasons, and so on. So the goal by several labs is to devise a chemical method where you can take two unprotected peptides, free peptides, with all the functional groups showing, and put them together, ligate them. And this is by Jimmy Tam, who was in our lab for many years, has moved down to Vanderbilt. And you can call it an aldehyde capture method. But the objective to get two big molecules to react is to get the amino group very close to the carbonyl. If they're in solution and they're flopping around, they're not close together, and they only occasionally collide in a productive way. But if you can hold this amino group very close to the carbonyl, then you can get a very high effective concentration. So the way Tam did it was to make a glycol aldehyde ester over here, and then react that with an N-terminal cysteine containing peptide. So the carbonyl would react with the SH and the NH2 and form the thiazolene ring over there on the right. And by doing that, you bring the nitrogen group very close to the carbonyl, and in the transition state of that reaction, you have these two 5-membered rings. So that was the goal, and he actually has been able to do that and to put sizable peptides together. Not yet a big protein, but so this is a new approach that's being used by a lot of people. You can also do this enzymatically. Next slide. This is using a subtleisen that's been modified in a couple of ways by genetic engineering, and you get what's called, I guess my light is burned out here, subtle ligase they call it. And here you can take two peptides, one of them must be an ester, and put them together, then remove the amino protecting group, R in this slide, and then you have another starting unit, and you can add the third peptide using that same enzyme to build up this peptide chain. Well, these people now, Jackson and Wells, I guess they're at one of the big pharmaceutical companies. Anyhow, they have made ribonuclease just this last year or so using this enzyme technique. So if it works as well in general, then it's going to be a very powerful new technique. People are also trying to do this with a catalytic antibody. The next slide will show that. This is work by Hirschman and Binkovich and their groups. So what they've done is design an antigen shown at the top, which has certain features that will produce an antibody which should recognize the two substrates shown below. One is a nitrophenyl ester of valine, and the other is a tryptophan amide. And it turned out that it did. The antibody would catalyze that reaction, form the peptide bond, and they were able to make a dipeptide. This has a long, long way to go to make it general so that a very big molecule can be made, but it's a first step. So I think it's important. So that was what I was going to say about proteins, and I wanted to go back now to a more current problem that we're doing in our own lab, and the next slide would show that. No, it isn't all I want to say on proteins. This is, one of the other efforts by a number of people is what they call de novo synthesis. So what the objective is to design a molecule, not the structure of the natural one, but one that will have the proper physical features which will allow it to act like the natural one. And this is something that John Stewart and his people devised. They decided to try to make a molecule that would act like chymotrypsin. And what they did was to make 4 separate peptide chains, all attached at the far end there, all held together with a network of lysine and ornithine. So you then have this base on a resin, and then you build up these 4 peptide chains containing the known functional groups that are present in chymotrypsin. So they did that, and they made a hole in it so the substrate would go in, and they made the hydrophobic groups in such a way that the whole molecule would stabilize itself and so forth. And it had enzyme activity. It was quite remarkable. So this has yet to be really confirmed in other labs, but when it is, I think it'll be recognized as an important step. Well, now I think I've finished with the proteins. Let's try this next one. Oh my. There we are. Okay. Now this is glucagon, which is a peptide hormone. And a lot of you I know a lot about diabetes and insulin and glucagon too. Anyhow, in the islets, in the pancreas, you have cells that produce insulin and glucagon and somatostatin. And those all interact with each other in various ways, either inhibitory or stimulatory. The glucagon, when it's produced, then will go over to the liver, where there's a receptor on the surface, on the lipid membrane, and it will bind, and then through a G protein it will eventually activate adenyl cyclase, and that will catalyze the conversion of ATP to cyclic AMP, and then eventually you produce glucose, goes out into the blood, so that raises the blood glucose level. So we wanted to make glucagon and study it, but the real objective was to try to make a glucagon antagonist, a competitive antagonist, and to diabetologists, this is a very naive approach, but anyway, our idea was that if you could block the inherent glucagon action, then in a diabetic, he would not need as much insulin to regulate the glucose level. So that was our working hypothesis, and so we set out to try to make an antagonist. The glucagon itself that we made was fine. It had all the proper physical measurements and biological activity. So the next slide then will show the synthetic procedure, I think.
We have an alternative we
Well, okay, there it is. So what we did, it's just the way I've been describing. So all of these peptides are made by essentially the same method. You take a resin that had a new kind of a linking group, and then build up the peptide chain stepwise from the carboxyl end until you've assembled the whole molecule. So that was okay. Now we had some leads, and we developed some leads that we thought would eventually produce an antagonist. It was known that the histidine in position 1 could be omitted and retain binding activity. So it would bind to the receptor. Then we found by kind of a devious route that aspartic at position 9 was really essential. You could put any other amino acid there and it would bind to the receptor from the liver membrane, but none of those would have any glucagon activity. So that separated these two functions, the binding function and the transduction function of the signal. And that was really the key to getting the antagonist. We also found an amide at the carboxyl end was helpful. So then we made that new molecule, or several of them, and the next slide shows the one of those. Yeah, okay, that's it. It's a binding curve, and for using the receptor from the liver membrane, and up the vertical axis is the iodinated glucagon, and you displace that or compete with that with your analog, and it shows concentration along the bottom. So our antagonist, the bottom one, the middle curve, then would bind almost as well as glucagon itself to this receptor. And then the next slide will show the activity. There we are. So we're measuring cyclic AMP, which is the end point of the hormone action, and across the bottom is peptide concentration. Well, this here and here. So the curve that goes up is the glucagon activity curve for cyclic AMP. The bottom curve that's flat is the activity of the antagonist. So it had no activity out way out. Now, if you then take a high level of glucagon and mix with it increasing amounts of the antagonist, then that right-hand curve with the dark circles, you see, it goes down, meaning the more antagonists you put in, the less activity you get out of the glucagon. So we got an inhibition ratio of about 12 in that experiment, and a PA2 value, which I don't need to go into there. This also, in the next slide, shows the blood sugar assay. If you use a lot of animals, rabbits, you can smooth out these curves, but we couldn't do that, so we only had a few animals. Anyhow, the top curve is the production of glucose by glucagon with time. So 10 or 15 minutes, you reach a maximum with intravenous injection. The center curve is if you put in a ratio of 50-to-1 of the antagonist, then you cut the activity down to half, and 100-to-1 it was virtually completely inhibited. So that that came out the way we wanted it. And the next slide shows the diabetic rat, and it, right on the left-hand side, the glucose level is way up from the normal, which is down at 100, because it's now diabetic. And if you then give a bolus of the antagonist plus an infusion of it over a period of several hours, actually, this blood glucose will drop and maintain more or less constant level. So this was good news. It had in vivo activity then. And the next slide shows an improved antagonist. Let's see, it's the bottom line, where we've made 2 more amino acid changes. We've eliminated serine at 11 and 16 and replaced it with serine, with alanine. And now this is 10 times better than the previous one. The inhibition index is less than 1, so that looks like a promising antagonist. We do have a little company who showed some interest, and we've given them this material, so we have some hope that they'll examine it carefully, and maybe it will turn out to be useful. Now, I have another project we've been working on that I just wanted to show a couple of slides. We've been looking at and I'm sure you can hardly see this, but it's a peptide from the silk moth, which turns out to be a good antibiotic. And what we've done is made a lot of analogs, and then we've made hybrids of the first half of the molecule is the one from the silk moth. Second half is the one melatonin from the honeybee. So we've made a composite molecule with 26 amino acids, and it turns out that's a very active antibiotic on both gram-negative and gram-positive organisms. And we also went ahead and made the D enantiomer, so the all D peptides, and in this assay, this antibiotic assay, they're just as active, fully active with the L. Of course, their half-life will be very much more. They're quite stable, being a D peptide. The next slide shows one application. You know, we keep hearing about the resistant strains that are developing, and tuberculosis is one of them. And this shows not the real organism, but a inactive variant, this Mycobacterium smegmatis, something we can handle in our own lab. And it has rifampin is kind of a standard antibiotic to use, and you can see if you we have 2 different assays here, but if you go down the last column, you'll see that the cecropin, L-cecropin was 20% as active, and the D-cecropin a little more than 100, and go down 3 more compounds, and here we have a cecropin A 1 to 8 and a melatonin 1 to 18, and there it turned out that's 3 or 4 or 5 times as active as the rifampin itself. So arrangements have been made to work in a laboratory also at Rockefeller, where they have the facilities to use the virulent strain, and we have some hope that we'll get those assays done pretty soon, and that this may turn out to be a useful kind of antibiotic. Very expensive. If you wanted to treat somebody with this, it'd cost a fortune. But you know, things like that change, and the cost can come way down. So this may, if we're lucky, turn out to be useful. All right, the next slide, I guess, goes to the vaccine. Yeah. We've also heard about vaccines and their value, and this is experiments on malaria vaccine. So it's a totally synthetic material, And this again was done by Jimmy Tam when he was in our lab. And he invented a new support which is similar to that thing I showed you before on the synthetic enzyme. So he has a lysine network at the carboxyl end, and that allows him to build up either 4 or 8 chains of peptide while they're still anchored to the solid support. He also, by synthesizing a lot of peptides, deduced what a B cell epitope and a T cell epitope would be, and so he's taken those now either individually or in combination onto this this support that he's talking about. And now all of these were used as antigens to see what kind of titer, and the next slide shows the titer he got. So with the one, whereas the B and the T and 4 copies, you see it's 50% way out at, this is times 100, so it's 25,000 dilution, roughly, something like that. So it's quite a high titer. We also heard yesterday about Paderoya's experiment. Tam had made this earlier, but he had not been able to do field tests of any kind. Paderoya has now come along and made not a branched one like this, but a linear one, but containing both the B and the T cell epitopes. And he's able to make it in large amount and has tested it in Colombia and Venezuela, and then recently in Africa. And these are ongoing experiments, which you heard briefly about yesterday. And the next slide shows a summary of some of the data. The effectiveness depends on the age group, and what he's measuring here are these episodes that you get if you have malaria, and he's comparing the vaccine with the placebo. So at the very young age, then it was reduced 77% effective, which is a good high number. For the intermediate age groups, it's only 20 or 30%, which is, a lot of people say, is not enough to be useful, but I'm not sure about that. And then at the higher age group, it went back up to 67. Manuel is a very enthusiastic man, and he gets a lot done, and he's pushing hard, and a number of other field tests are being done. He also has a second generation antigen now, or vaccine, which he is very encouraged by. So I think it's entirely possible that this synthetic malarial vaccine will become of considerable importance. How am I doing it? So I'm going to run out here in a minute. So I had a couple of other things. The next slide shows this is a peptide library. Another big push. If you want to make a lot of molecules and screen them in some way, then making these combinatorial libraries is the way to go. This particular one that Kit Lamb devised allows you to make one peptide species on one polystyrene bead. You have lots of beads, and they're all mixed in together, but any one of them contains only one peptide sequence. So what you do, and I can't go through all this now, but you get every combination of the whatever number of amino acids you put in. Every combination will be represented, and you take the beads and spread them out, and for example use a fluorescent antibody, and the ones that light up will be the ones that have the activity that you're looking for. And so you then take a pair of tweezers and reach in and pull out this little bead, and you do a micro sequencing on it. Now you know the sequence of an active peptide. So then you go back and make a larger quantity by the standard method that I've been talking about before. And I guess every drug house in the world is doing this now. And they're using not only standard amino acids, but also mimetics of all kinds in the hope, in searching for new drugs. So that's what they're doing. They can scan this vast number of peptides and hunt for a structure which will give them a new drug. Houghton found, for example, he found an active peptide which was made out of all D amino acids, but otherwise no resemblance to the native structure at all. So you can come up with things you can't predict. Really, you can't predict them, but you can screen for them this way. So I think it's a very exciting area that's obviously going to be expanded a lot. I think I had one more slide. Oh, this is PNA. I obviously am running out of time. I think I better just not cover that. But PNA is a combination of a nucleic acid and a peptide. They've removed the sugar phosphate from the backbone and replaced it with a peptide chain. So again, this is peptide synthesis, and these things will bind to the DNA tighter than the DNA duplex. Will hold tightly. This will displace one of those chains and come in instead. And my friend at work has used it in a very neat experiment to isolate active genes, for example. But I can't cover that, and I think there are lots of other examples, so I think I'll just leave it with this number now. Thank you very much. Anybody? Yeah, there's.
I'm interested in juvenile diabetes, and I'm wondering if you said that a company had shown an interest in your research on glucagon. I'm wondering if that has possibly implications for a pancreas that will not be, cannot be restarted again. Will it just lower the amount possibly of insulin that needs to be taken?
That's the idea at this point, that it would simply compete with the endogenous glucagon and therefore reduce the amount of insulin required. Yeah.
Would that interfere, or would that change the dietary requirements?
I don't know. Really, we don't know. I think the ketoacidosis will also be affected, and maybe an even easier area to work in. And a couple of people have said they thought there's a real need for such a thing now. So they're going to do some of those experiments too. This is all right at the very beginning. Okay. All right. Thank you.